dear student, today we will cover another topic of genetic engineering from course code RBT 501 of third year, and it is the topic is from unit 3, and myself is Dr. Deepak Gola, assistant professor in the Department of Biotechnology, Noida Institute of Engineering and Technology. So today we will cover about the interaction about the protein and nucleic acid interaction, how we know that interact with the nucleic acid. So the protein can be anything like hormone, enzyme, or we know there are two type of nucleic acid in our body. It is first the deoxyribonucleic acid, that is DNA, and second one is ribonucleic acid, that is RNA. So or any other protein will interact with this DNA or with RNA. So there are two basic techniques which we will learn in these slides. So first of all, what is the need of study of these DNA interaction with the protein or RNA interacting with protein? So why it is important? It is due to the fact that nucleic acid carry genetic information, and that provide and that protein, these protein regulate various life processes. They are considered to be the part to be the two of the most important biomolecule in the living organism, because these protein will interact with DNA or RNA, will make another proteins or signal for various life processes like growth, or for another stimuli about provide the immunity against some signaling molecule, or bacteria is coming to our cell, it provide the signal processing. Then their interaction play a crucial role in most biological processes, which include everything like replication of the DNA during the cell cycle, then transcription, translation for making the proteins, and recombination of enzymatic event, like in the last topic we will learn about the signal processing, where the number of protein will interacting and leading to the some type of these protein are interacting to give rise to some other processes. So here is the example of protein interacting with the nucleic acid. The first we all in the previous classes we learn about the how DNA will replicate. So in this how DNA replicate, it will make use of a enzyme called DNA polymerase, and it will interact with DNA to make the multiple copies of the DNA. Thereafter after replication we learn about the transcription. In transcription, what is transcription? DNA will make a messenger RNA, and with the help of what? RNA polymerase. So this is a type of enzyme, and we know that all enzymes are protein, but all protein are not enzymes. So these enzyme will interact with this DNA to make the copy of messenger RNA. After that, the messenger RNA is transcribed, it will translate into protein. It will translate into protein with the help of some enzymes. And what are enzymes? Enzymes are protein, like translocase enzyme. So these are the example how protein will interact with the nucleic acid in our day to day life. Okay. So there are some method to analyze how the protein will interact with the nucleic acid. So how to analyze them? There are two type of assay we know we will learn today. That is, first is IMSA or gel shift assay. What is IMSA? IMSA is electrophoretic mobility shift assay, and second is DNase footprinting assay. This DNase denote enzyme. Which enzyme? DNase enzyme. So, how will you recognize that something is some you will get to know about what is enzyme or which is not enzyme or which is enzyme. So, most of the, like in 90% cases, enzyme name contain last word is. So, these protein we will isolate the protein and we will incubate that protein with our DNA or nucleic acid or like RNA, and we will learn that this protein that we have isolated will interact or will not interact with our nucleic acid. So, first of all is the first technique is the DNA footprinting. So, first of all we require the protein, we will require a protein, and the second thing we require will be the our DNA, our DNA. So, what we will do? We will incubate both these and will incubate for some period of time, and it will be like providing a optimized condition. The protein may now the two cases will arise after incubation. Two cases will arise. The protein may or may not interact with the DNA. So, these are the two cases. It is not interacting with the DNA, the protein, and protein is interacting with the DNA. So, we can draw it like this. This is protein, and in this protein is not interacting, in this protein is interacting. In this it is not interacting, not interacting, and in this it is interacting. Now, we will treat these both the sample with DNase. DNase is enzyme which will cut down the DNA at various site. You also know this is also know that this is our residue level. This is both are residue level at 5 dash end. Which end? 5 dash end. This is 3 dash. This is 3 dash. So, with the use of enzyme, let it be of different color, might be of yellow color. We will take yellow color. So, what this DNase enzyme will do? It will cut down the DNA from various multiple sites. So, here also it will try to cut down the DNA from multiple sites. So, one thing is important, when enzyme is interacting, this site will be not available for the DNase enzyme. So, it will not cut down, it will not cut down from this site. So, this site will remain intact, this site will remain intact, this site will remain intact. So, this site will remain intact. So, now we will run gel from it. This is our gel, these are the well. In first we will put the sample from here, and in second we will put the sample from the one in which DNA is interacting with our protein. So, now we will run the gel provide the voltage and it will run, and we will get the different okay, it is something So, the longer sequence will be on above and shorter will be below. So, what we will get to know that some of the band are missing. Some of the band, for example, this and this, you will see that some of the band are missing in your gel, like this band is missing. So, why it is missing? Because the DNA, our protein bind with that site and DNAs will not cut this site, and this fragment will remain intact and it will remain missing from our gel. So, from this we will analyze that this is the site, where DNA is interacting with our protein. So, we will find with the help of radio label, these are radio label. So, we will find from and that at which sequence that our protein is interacting with our DNA. So, this is the overview of the project. As you can see, this is radio label DNA. We will take DNA will make the cut down the fragment into various part, and this is with the protein. Protein will bind with the DNA and the site will not cut down with the by the DNase enzyme, and these part will be missing. This part will missing, this one, this part will remain missing in the gel also. So, we will find that from the end we will can analyze that at what base pair the protein is interacting with our DNA. So, this is all about the DNA footprinting. We will move to the next slide about these are the result or the conclusion from the gel electrophoresis we can make out, like the site bind to the protein will not be nicked by the DNA enzyme. So, why? Because that site is occupied by the protein. Next is some band will not appear on the gel as compared to control. Why? Because the DNA enzyme will not cut those fragment. Why? Again, because protein at that site is interacting with our DNA. Next is the fragment not appeared in the gel are the place of protein binding. So, we will move to the next technique, that is IMSA, or we will call electrophoretic mobility shift assay. It is also called, dear student, is also called affinity electrophoresis technique, protein DNA gel shift assay, gel mobility shift assay, band shift assay. So, for this we do not require the DNase enzyme, we only require the DNA fragment and purified protein for this, and gel electrophoresis. We do not require radio label also in this case. So, what happened in this? It is like similar to our previous case. We have protein, let it be in circular form, then we have our DNA, this is our DNA, and we will incubate these, So, there are two cases, in this also there will be two cases, either the protein will interact with our DNA or protein will not interact our DNA. So, this is the case, it is will interact and will not interact. Okay. So, we will draw the protein also with the red color. So, it is not interacting, here it is interacting, here it is interacting. Now, what we will do? We will again run the gel electrophoresis for it. Let be gel of another color like blue color. This is our gel, these are the wells. So, we will put the sample from both the thing, both these, because first this and from second is this, in which the protein is interacting with our DNA. What you will find, student, you will find after running the gel, you will find that DNA in which protein is not interacting with our DNA, that this sample will move towards end, where the protein, where in another sample where the protein is interacting with our sample, will find there is another band which is below or above in the gel. It will above the sample, above the DNA band. Why it is like this? Because when the protein will interact with DNA, it will make this sample, the molecular weight will become on higher side. So, when the molecular weight become higher, it will become heavy. So, it will take time to move. It is as simple as that, like if a person is moving alone, it will move very fast, but if there is a like a sack of grain on it, it will move slow because it is feel heavy. So, it is same like that, it will not move. So, why this is the second band? Because we will find that not total protein will not interact with the total DNA. So, we will find this band also, So, this is IMSA, and this is also called retardation. Why retardation shift assay? Why? Because in this our band is retarded, it do not move as much as compared to the control, in which DNA is not interacting with our protein. So, that this is the overall neat and clean diagram that we have learned in above. So, here we can see the protein is interacting our DNA, and this is the binding reaction, these are buffers conditions. So, here we can see this is the movement is retarded. Why it is retarded? Because the DNA become heavy after interacting with protein, the molecular size of the DNA is increased. So, it will not move as fast as compared to the simple nucleic acid, which is not interacting with our protein. So, what are the conclusion from these gel electrophoresis of IMSA? We can find that binding of protein with the DNA will make overall complex heavy, and the DNA protein complex will move slowly, the movement will retarded as compared to the DNA or nucleic acid alone. And the next is DNA protein complex band will appear above the DNA unbounded protein in the gel. So, that is all. So, we will today we learn about the two techniques, IMSA and DNA's footprinting assay. This is the basic about the protein, how we will analyze that protein, isolated protein is interacting with our nucleic acid. So, thank you students, and if you have any query, you can contact me on deepakgola@gmail.com. Thank you once again.